Hi all, welcome. This seminar is going to be given by Professor Rajeev Prabhakar, who is Professor of Chemistry at the University of Miami. Dr. Prabhakar has a PhD and a postdoc in quantum chemistry from Stockholm University and Emory University, respectively. He applies computational approaches, including pure quantum mechanical, hybrid quantum mechanics, molecular mechanics, so QM, MM, to investigate reaction mechanisms catalyzed by both enzymatic and non-enzymatic systems. And he has published more than 95 journal articles on this topic. And today he's going to talk about applications of computational chemistry in the development of catalysts and materials. Thank you very much, Dr. Prabhakar. The floor is all yours.
Thank you. First of all, I would like to thank Dr. Surinani and Barde for the kind invitation. And I'm glad to be here. And just give me 1 second. All right. Okay. So, oops. All right. So regarding the field, I mean, the field called like theoretical or computational chemistry, it started in 1920s with the famous statement given by Paul Dirac. And according to him, the fundamental laws necessary for the mathematical treatment of a large part of physics and the whole of chemistry are thus completely known. And the difficulty lies only in the fact that application of these laws lead to equations that are too complex to be solved. And it's a very bold statement for 1920s. And there were no computers. And now, not many of us, like people know that he did actually his sabbatical in physics department at UM. And now we are much closer than ever to actually realize that goal. Like, so what he said that, okay, if you solve some equations, you can probably explain most of chemistry and physics. And if for Christmas, if you are more interested, you can read his biography. It's available on Barnes and Nobles, called The Strangest Man. And he was a very remarkable scientist. He was father of quantum electrodynamics. He gave Dirac equation. He predicted antimatter, string theory, and magnetic monopoles. And he got Nobel Prize in 1933. So this is my only slide with any kind of equations, I promise. So basically in the field, we solve two equations, as Dirac said. One is we use Newton's second law of motion. As we all know, this is high school physics, where we have force is equal to mass multiplied by acceleration. And we use this equation to predict structures of macromolecules like proteins and host gas complexes and protein interactions. The second equation is this innocent-looking equation called Schrodinger equation. But people who work in the field know that this is probably one of the hardest equations to solve. And there have been many different methods to solve this equation. And for that, Walter Kohn and John Pople, they got Nobel Prize in chemistry in 1998. And in this equation, this is H, which is Hamiltonian, which represents the sum of energies of the system. And it operates on this wave function, gives us energy and the wave function, right? So how do we do that? And then again, there was another Nobel Prize in the field in 1913, like few years, 80 years ago. And then it was given to Martin Corpus, Michael Levitt, and Arya Warshall. And this is, by the way, statement came from the Nobel Committee. And according to them, the chemists used to create models of molecules using plastic balls and sticks. Today, modeling is carried out in computers. So in 1970s, Karplus, Levitt, and Warshall laid the foundation for powerful programs that are used to understand and predict chemical processes. So computer models mirroring real life have become crucial for most advances made in chemistry. So today, the computer is just as important a tool for chemists as the tube. Simulations are so realistic that they predict the outcome of traditional experiments. So the goal of this talk is that at the end of this talk, I should be able to convince you that it is correct. And I will try my best. So let's start with some basic interactions. So first is we start, like, as we know, there are some fundamental interactions in nature. And these interactions actually lead to the, creation of molecules and materials and everything in chemistry. So first is metal-ligand interaction. So as you can see that metal can bind to different ligands in many different ways. It could be in linear form, trigonal planar form, tetrahedral and square planar form, and so on. And of course, it varies depending on the electronic nature of metals and these ligands. So there are many, many different, structures available. And another is like basic non-covalent interactions that are very important in chemistry. So for instance, interaction between two charges that is inversely proportional to distance between two charges. A charge can interact with dipole, two dipoles can interact with each other, and so on. And as you would notice on this in this column that all these interactions are distance dependent. And depending on that, their strengths are different. All right. And another widely very common interaction is hydrogen bonding. So this is just like I'm going through the basics. Maybe I think some of you probably took chemistry a while ago and might have forgotten these interactions. But I would like to convey the message that these basic interactions, they play a very critical role in chemistry, and they lead to like creation of molecules and everything that we work on. So hydrogen bonding, for instance, in hydrogen bonding, you could have A, which is an electronegative atom, and hydrogen is bound to it. So what happens then, since it's higher electronegativity, there is a positive charge on hydrogen, and that interacts with the lone pair on the second atom. So that's your hydrogen bonding. Then we have hydrophobic interactions, where that allows the mixing of polar molecules with nonpolar molecules in polar solvent. And what they do, like, so it's driven by entropy, where nonpolar molecules, they are surrounded by polar solvent, and then just to confine them in space. So these are hydrophobic interactions. And also we have some repulsive and attractive interactions, where if two electron densities, they're far from each other, first there is attraction between them. And then there is an optimum distance at which they stay, they gain energy. And if they come any closer, it becomes repulsive. So these are kind of van der Waals interactions. So if we combine all these interactions and some more, we create molecules. And how do these molecules interact with each other? So in traditional host gas chemistry, there are two cases. One is this is our guest, all right? And this is the cavity of the host. As you can see that this is very rigid. You can't change it. But then there is a host gas complexation take place, where this gas molecule sits very nicely in the space provided by the host molecule. Now, in biology, actually, so the host gas chemistry is a little bit different. So this could be our guest, and this is the cavity of the host. As you can see, that guest is much bigger than the space provided by the host, but they also make this complex. So in this case, the dynamics and plasticity of both host and guest molecules are very critical. So, and then in both cases they work, it just depends on the nature of molecules. And also when we want to form material, as you can see that you can start with a small molecule, and then they interact each other with through non-covalent interactions. They can form this higher order oligomers, fibrils, and then you can call them materials, and and all different kind of chemical forms that we see in nature. So I will give you an example of an enzyme. So we have worked on many different enzymes, but this is a good example. And then it's called, so GPDQ stands for glycerophosphodiesterase. So as you can see that when we use those interactions, metal binding and non-covalent interactions, we form proteins or enzymes like that. And in these enzymes, this is the active site of these enzymes where the reaction takes place. So that's here where the bond formation and bond breaking take place. And it has been a long-standing goal in chemistry to design small molecules that can mimic the activities of these enzymes. So since mid-1990s, it has been one of the holy grails of chemistry, and it's still no closer than we were 25 years ago to make these molecules that can mimic the catalytic activities of like normal, natural enzymes. So as you can see that these are simple peptides made from very small, few amino acids. These are these are dimeric. You could have either 2 or 3 of these. You can have trimeric strands, and you can have tetrameric bundles. I will go through them later. So, and then these catalysts, so these are used to catalyze two most fundamental reactions in nature. One is peptide hydrolysis. As some of you would remember that in nature we have 20 different amino acids, and they all form our peptides proteins through the formation of this amide bond or peptide bond. And as expected, this bond is extremely stable, and its half-life is about 350 to 600 years at room temperature and neutral pH. Now, there's another type of bond is phosphoester bond, which is used actually in our DNA and RNA. And this bond is even more stable than the peptide bond, and its half-life is about millions of years. So that's why it's used as in genetic material. However, the break cleavage of those or splitting of those bonds is very important in most of biology. For instance, in control of cell cycles, cell death, DNA repair. And due to these biological functions, these reactions are connected to probably 75 to 80% of diseases, such as HIV, Alzheimer, malaria, cancer, diabetes, hypertension, etc. And also in biotechnology, if we want to design a new protein, we need to create long sequence of amino acid residues, which is created using these reactions. And it used in proteomics, genomics, therapeutics. And therapeutics is interesting here because they are about few hundred neurological disorders. And all these disorders, they are associated with the aggregation of a specific peptide. So if we can figure out a way to disrupt them, that would lead to the treatment of a lot of these diseases such as Alzheimer, Parkinson, mad cow disease, Huntington, and so on. And also bioremediation of pesticides, I will talk about, and nerve agent, I will talk about it a little bit more. And it turned out in industry, about 60% of all industrial enzymes are proteases or peptidases, enzymes that catalyze these reactions. And they are used in textile, food, leather, paper, bioethanol production, and so on so like in wide range of industries. And here, just a fresher. So what I will do, if you have forgotten the names of your amino acid residues, so that's okay. And then also, it's just the name is different. So here is the arginine. What it does, it's a hydrogen bond donor, all right? Then there is a histidine, which is a neutral residue, and it binds to metals. Then there are aspartic acid and glutamic acid, and then as you can see that they are negatively charged carboxyl groups, so they are hydrogen bond acceptor, all right? So I will just mostly focus on these residues, all right? So hydrogen bond donor, residue that binds to metal, and also hydrogen bond acceptors, all right? So let's start with this enzyme. So in nature, there are three types of monoesters bond, phosphoester bond. Monoester, as you can see that there is one OR group, diester, which is actually in our DNA. So there are two OR groups, and triesters, which have three OR groups. Now, there are many different types of, you can change these molecules by just changing this R group or R prime, all right? So phosphodiester bond, this one is natural, it's in our DNA, but triester bond, it's man-made bond. And it's found in pesticides, insecticides, and all that. And then in nature, since it's a man-made bond, we don't have natural protection against it. So that means like we humans, we have no way to treat if we get these chemicals in our body, and we can't get rid of them. And that's why these days Trader Joe's and Whole Foods, they're very popular because everybody wants to buy organic food, where these insecticides and pesticides are not present. And also take a step further, and then many nerve agents like mustard gas, and then this Novichok, which is very popular these days, they all contain phosphotriester bond. And that's why we find it very, very hard to get rid of them. We don't know. We have no protection against them. So this enzyme, what it does, it's promiscuous. So in chemistry, the promiscuity is a very good thing. That means that one molecule can catalyze multiple reactions. And this enzyme, GPDQ, abbreviated form of it, can hydrolyze actually all three types of bonds, and even some organophosphates that are used in nerve gases. So it's a very important enzyme. And the way it is created, so it has this alpha site where alpha is iron. And when the substrate comes, when the molecule that needs to be degraded comes, then it creates a beta site, right? Otherwise, it stays in inactive form in this iron bound form. And then what happens? It completes the first coordination shell, and then there are 6 loops. These are formed by secondary coordination shell residues. They don't directly participate in the reaction, but they do stabilize. And then they interact with these substrates through non-covalent interaction. And then they are very flexible because they can create space or they can shrink the space depending on the nature of the substrate. And then we have some other like water and then other residues that form hydrogen bond and complete this second coordination shell. And then this is called breathing of the active site cleft, where depending on the nature of these molecules, we call it substrate, they can adjust and then the reaction, they can be degraded. So when we start with this reaction that it's in monomeric form, now substrate comes in, as I told you before, that it creates the second binding site. And then the second metal comes in and enzyme becomes active. So we wanted to study how this process takes place. All right. And what we did, so we created structure, and nobody knew actually, because experimentally you cannot follow this process. And so we created structure of this mononuclear form, substrate bound mononuclear form, and binuclear form. And then we got their structures. And then what we did, we started going backwards. We started removing them one by one and see that if you can go backwards even. And then what did we learn from it? So we got these tables. And then what do these tables represent? We can calculate binding energies actually from this. And then, so what these computational simulations do, they provide you structures as well as energy. Okay. So we learned that, okay, so then the binding of the substrate, it enhances the metal binding affinity at the both alpha sites and prepares the beta site for the coordination of the second metal. And in the binuclear form, this one, the active form, the binding affinities of both metals increase. So that means it's a favorable process. But if you remove the substrate, and then there is a reduction, so that means you can go backwards also. So now we have this structure, and now, as I told you, that this enzyme is promiscuous. So what it does, it can degrade all these different types of molecules. And then, so for instance, these, as you can see that based on, if you even look at their overall shape, so red color, it represents the negative charge, and blue color, positive charge. As you can see that their charges, where they are localized, and their shapes are quite different. And by the way, this peroxide, this third one, it's a very common insecticide. And what this, and now since it wasn't known that how do these different molecules interact with this enzyme, we use our computational techniques and try to study their interactions. And so what we learned, as you can see that these colors, as I mentioned, so red color represents the negative charge, blue color positive charge. You can see that depending on the nature of these molecules, these inside even in the enzyme, these colors change. So like that means there is a dynamics involved, which allows this enzyme to accommodate all these different substrates. And these 2D interaction graph, it tells us what kind of interactions are there when these different molecules are bound there. And then we can get these structures. We can also look at energy based on how many different energy states it went through as it was binding to this enzyme. And also we can look at this coordination flexibility, which I refer to as the breathing of the active site cleft. And then, so what these loops, they form an irregular hexagon. And then we can calculate its volume depending on the nature of the substrate. And then we can compare what exactly is happening. So we did that for all these 4 different substrates. And then what did we learn from it? So the chemical nature of substrate, it influenced the mode of binding, electrostatic surface potential. This is electrostatic surface potential, as you can see. And then metal-metal distance and reorganization of the active site. All right. And then it also went through like different energy basins, like different energy states. So let's focus on this. So you can see that here. You see this is principal component analysis. So monoester, it looks very different. Then for diester, this is another diester. And this is triester. As you can see that it changes. So they go through different energy states as these molecules go inside the enzyme and bind there. And also we calculated the binding free energies, for instance, here. And then we can see that mono and diester, they bind with comparable binding energies, but the triester, which is peroxide, it binds with much lower binding energy. All right. So now we have the structures of these molecules. Then how are they degraded? And so there are 3 proposed mechanisms. Like one is that, so this is, and they use water, by the way, to degrade this. So it's a hydrolytic reaction. And you can use this R2 as hydrogen, then it becomes water. So water attacks, and this we break this PO bond. We can break this bond with even before water attacks. So that is called dissociative mechanism. And in nature, we have never observed for in any case, there is no dissociative mechanism. It could be a concerted process. As water attacks this phosphorus, this bond is broken, as you can see here. And it could be associative as well, where this water comes in the form of hydroxyl and form this intermediate, and then that bond is broken. So now the question is, which mechanism is favorable here for all these different molecules? And we studied using these models of active site for all these cases. So now, without going into details, you can see that these arrows, they represent how proton moves, right? So for instance, in this case, this could be a stepwise process where we go through an intermediate, or it could be a concerted process where we can do everything in one reaction step. And we studied these mechanisms for all these different molecules. And then what did we learn in the end? And so this is typically in the field called potential energy surface diagram. So what it represents, this is your reactant, because from these quantum mechanical calculations, we get energies and structures of these molecules. And when we put them together, it becomes potential energy surface. As you can see that the colors represent, if this is the GP green color and the black color, they are diesters. Blue color is triester, insecticide, and red color is monoester. As you can see that they go through different energy barriers. And if you take this activation energy and plug in Arrhenius equation, you get rate of reaction, and that one can measure using kinetic experiments. So based on that, you can tell, first of all, we learned that if the reaction takes place in one step or in multiple steps, the energies, the barriers are quite similar. And other thing is that there are clear distinction between the barriers or the rate of reactions for different substrates. So it is very clear that this enzyme is more efficient in splitting or hydrolyzing a diester bond than mono or triester bond, right? And that's what we learned from these calculations. And now I will just switch gear and then let's talk about more about material, because this is School of Engineering. And so the amyloids is when these peptide comes and then they form these higher order structures, and they could be the good, the bad, and the ugly. I will start with good first, and then we will talk about bad and ugly. So one is they can form catalytic scaffold, like these simple molecules that can catalyze reaction, and they can also form biomaterials actually. So I will start with this simple molecule. It's called clavinin. So, and it's found in is a sea squirt. It's called Styla clava, and it's an antimicrobial peptide, and it actually looks like calamari. So you can buy it and eat it. And this molecule contains this simple, this Styla clava contains a simple molecule called clavinin, and it looks like, so it has hydrophobic side and hydrophilic side, all right? And when we eat this, or when it goes in our body, so this is the outside the cell structure. And then it can sit on the membrane, and it could go inside the membrane, and it can bind zinc. And when it binds to the zinc, it can degrade, break the DNA of many pathogens. So there are, according to NIH, there are 8 pathogens. They categorize as escape, ESKAPE. And we propose a mechanism that, and found that, showed that this actually simple molecule degrades all those 8 pathogens in cells. And here, so if you stack these peptides, they can form these simple structures. As you can see that here, these the blue dots, they are metals, zinc, and then you have metal binding here. And then if you look at the bottom, these are all hydrophobic interactions. So they can form scaffolds like that. And only thing we need, and they are formed from these simple peptides. It has only 6 amino acid residues. And then if you tinker the 6th one, which is this is hydrogen bond donor. This is hydrogen bond, it's hydrogen bond acceptor. This is hydrogen bond donor. So if you change one residue, their catalytic activities change. All right. And actually, we also looked at their material properties. How does it look? So when you have the peptides, when you stack them together, they form fiber like this. And you can also look at what happened when metal is binding here, like I showed here. So when metal binds, the overall structure remains the same, but there are some changes, because by looking at it, you can't really tell. And what we do, we can calculate their material properties like Young's modulus and bending rigidity. How do we do that? So that is equivalent of doing atomic force microscopy experiment, AFM experiment on computer. So what we do, we make these fibrils, And then we constrain the edges where deformities are, and then we apply stress in one direction. Then there is a strain in the opposite direction. And from the stress strain calculations, we can take the linear part of it, and then we can calculate Young's modulus from the slope of it. And as you can see that when there is no metal bound, the Young's modulus of these fibrils is 6.2 gigapascal. But when metal is bound, it actually gets lower. It is 4 gigapascal. And also we looked at other structural properties, like for instance, what is interstrand distance between these molecules? What is the fibril twist? It's like a rope. If you elongate a rope, it starts to twist to provide stability. So we looked at the fibril twist as well. And we looked at interstrand angles, and then periodicity, and then Young's modulus. So what is interesting to see here, as you increase the size, the Young modulus actually start decreasing. We can look at, like, if you have really small rope, it's very hard, but as you elongate it, so it becomes a little bit softer. As you can see that here, the Young's modulus at some point, then it converges. But these fibrils, when, as I said, that we use them as catalytic scaffold, if you look at only 2 of these peptides, monomers, they form one metal bound site. If you use 3 of those, they create a binuclear site where you have 2 metal centers, and they can catalyze reactions like enzymes do, like monoester hydrolysis, diester hydrolysis. Another thing I would like to show you, so just for clarity, we have removed the rest of this molecule. As you can see that, so here, as I told you, we get structures and we get energies of these molecules. You can see that, you can see in front of your computer screen that how protons are moving. So it's going from this water through this water to this molecule called histidine. And then when it goes there, this hydroxyl group attacks carbon. As you can see here, this hydroxyl group is attacking carbon and forming a bond. And then you can see that how this proton is transferred to break this bond, and then you can follow it, right? I can just tell you, experimentally, you can see all these structures because these reactions, this occur like at picosecond or nanosecond level. So there is no way to observe these transitions. And if we use, for instance, 2 metal centers, we can see that, oh, we can hydrolyze even a diester bond, okay? And we know the activation energy, which we can, use to calculate their rate constants. All right. One thing is, now this is the second generation of these peptides. As you can see that IHI. So the names are irrelevant. This is this isoleucine, which is a hydrophobic residue, which doesn't like to interact with water. And you replace this I with F, which is phenylalanine. So here you can see you have this aromatic group. This is also hydrophobic, but chemically they are different. If you form this scaffold or fibrils using this peptide, they look like this. If you use this one, they also look like this. So they all form scaffold, but this one can convert carbon dioxide into bicarbonate. Okay, so it can hydrate carbon dioxide, which is a major problem these days. This one also forms similar structures, but there is no CO2 activity. So it's not active, and this one is active, and we still don't understand why it's like that, because they both look quite similar. And this is like how CO2 actually hydration takes place. As you can see that here, this carbon dioxide binds to this oxygen, and after that a proton is transferred. So there are 2 different mechanisms. Lipscomb, actually, he got a Nobel Prize in chemistry. According to him, there is a transfer of proton through this water, and Linscu, who didn't get Nobel Prize, but according to him, there is a rotation of this group. And we looked at both, and it turns out that Lipscomb mechanism is more favorable than Linscu. All right, and now we will move to a little bit more complex structures. As you can see, these are trihelical strands. And what is the advantage of these trihelical strands is that they are hydrophobic from inside, as enzymes are. They are soluble in water, and then also you can crystallize them. And they could be either homo or heteronuclear. That means that they could be formed by the same strand, or they could be formed by the mixing of 2 different strands. And so I will give you an example. So this is strand A. So here, as you see that this residues in purple, this is alanine A and this C. So this is our strand A. If I interchange them, we form strand C. So as you can see that location 12 has C and location 16 has A. So we interchange them. However, now these are both hydrophobic residues that don't like water. So if we replace them with a smaller one, so alanine to leucine, then if we do the same thing on A, we form strand B. And if we do the same substitution in strand C, it forms strand D. All right. So what is important when we mix strand with A and B and C and D, what happens? They selectively form A2B and AB2, like heterotrinuclear strands. They don't form homo trinuclear strand, like, so you don't see A3 and C3. Now the question was why it's like that. And you can see that there's a lead NMR data, there are PK values available. So what we learned, we used our computational techniques and calculated their complexation energy. So we splitted them apart and then see that how much energy we gain by making them. And it turned out that A2B and C2D, they are energetically a lot more favorable than the other forms. And why it's like that? So it's just like, again, like I mentioned earlier, so what is happening? So there are three fundamental interactions in nature: metal binding, hydrophobic effect, and hydrogen bonding. They are playing critical roles. So if you look at for instance, the A3, you see that these three water molecules, they are interacting either with each other or with these sulfurs inside A3. If you go to A2B, they interact kind of similar way, but the strength of these interactions is different. All right. Here, I think a clear example is C3. In C3, you see that three waters, they form a triangle. They interact with each other. But you go to C2D, that triangle is not there, and they interact in very different way. So that means in a productive way, we can manipulate these fundamental interactions and make totally different type of structures, and they have different activities. We can also go to tetra strand, like these are the tetrahelical bundles. There you can create two metal center sites. So you can form from two different chains, or you can like even link them covalently, and then you can form just a one long chain, and you can fold them to four helices, and then you can form the same type of molecule. Or you can have four different chains and stack them together. So here what we did, we looked at the effect of metal. What will happen if we replace just metal in these? And this is based on the experimental results. I will show you. So if we have two manganese, for instance, and here you can see that how it's catalyzing the reaction. This is called transition state. So barrier is 28 kilocalories per mole. But if I replace two manganese with one zinc and one iron, as you can see that it goes up. It's actually worse than dimanganese form, 28 to 29. And then if you go to zinc, it's even worse. And that's also actually confirmed by experiments. And now I will switch gear. We will talk a little bit about drugs, actually, how you can use these techniques to design probes and drug molecules. So this is a brain of a normal person, hopefully us. And this is a brain of a person who has Alzheimer's disease. As you can see that the brain of a patient, Alzheimer patient, is characterized by these plaques. And these plaques are actually formed by a simple peptide called A-beta 40 because it has 40 amino acid residues, or it could have a longer version for A-beta 42, which has 2 extra hydrophobic residues. So we all produce these A-beta peptides, but some people get this disease and some don't get. And it all depends on when these molecules aggregate and then form neurotoxic structures, which can start killing our neurons. And now the question is, can we come up with molecules to understand when do they aggregate and then study their neurotoxic forms? And just to tell you that these structures, they don't form X-ray. They don't form crystals, so you cannot crystallize them. So you cannot look at their structures easily. However, in collaboration with Angel Marty at Rice, we have developed some probes. And then, so just a refresher, like, so, like it's it's interaction between light and matter. When you expose matter to light, light is absorbed, like in form of laser, for instance. You could have absorption, and then absorption is like this, so you give some energy to your matter, it goes to excited state from, for instance, S0 to S2. And then there are two possible outcomes. One is fluorescence, where the energy, because it's a high energy state, the molecule comes down to the low energy state and release excess of energy in terms of light. So this is fluorescence. Now there is another outcome where it can go to intersystem crossing. That means it can change from singlet to triplet state. and the energy is used in the vibration and rotation of molecule, and then there is a slow decay of energy. It's called phosphorescence, right? So here, so in collaboration with Angel Marty, he can do all these experiments actually. So what we looked at this ruthenium molecule, which is photoluminescent. Like, so what it does when it interacts with fibers that give us Alzheimer's disease, it becomes, there is a 40 times enhancement in photoluminescence. That tells us these fibrils are formed. And also based on the jaw plot, they could measure that this molecule, ruthenium molecule, interacts with about 2.6 monomers, like molecules of this peptide. What they didn't know, where does it interact? So we use our techniques and we propose that, oh, it interacts with on the surface of this molecule through these hydrophobic interactions. And what they did now, so okay, is that correct? So they made another molecule, peptide, that has same morphology, but did not have this binding site. And there was no enhancement in photoluminescence. They said, all right. Now we try to design a next generation of molecule. By the way, Jackie Barton at Caltech, like she studied the interaction of this molecule with DNA. So instead of these peptides that are involved in Alzheimer's. So this molecule was used before. So now we form this rhenium molecule complex, which has different ligand environment. And it is very remarkable, more remarkable that because it absorbs red light. Now, red light is very important because it can cross tissues and bones. And also this rhenium complex also interacted with exactly the same binding site as the previous ruthenium complex. And what it does when you shine, so you feed this, when you shine red light, what it does, it creates singlet oxygen. And when it creates singlet oxygen, it can hydrolyze, it can oxidize, different residues like methionine. So there is a sulfur, if you attach oxygen atom to it, becomes sulfoxide. You can take, for instance, histidine, you can oxidize that. And these residues, they are non-polar, so it's hydrophobic. But when you attach oxygen atom, you change its dipole moment and make it hydrophilic. So when we do that, we can selectively disrupt these fibrils. And so this is kind of photodynamic therapy for the treatment of Alzheimer's. And we also showed in mice, actually. So this is the mice model. As you can see that when we use the rhenium complex, all these fibrils they vanish, whereas they stay if we don't use this rhenium complex. And these experiments on mice were done by Ines Moreno Gonzalez at UT Health System. Now I have 10 more minutes. So fibril formation. So now as you know that, now we can take with a simple molecule, we can stack them, and then they can form this really different forms, like with different sizes and morphologies and all that. And it could happen in many different ways, like 2 of these could come together, 3 of these could come together, 4. So it's not very clear how do we reach from here to there, but we do reach, and very fast. So what they do, so now the big question is here, based on the structure of these molecules, can we predict their structure and their mechanical properties? And so what the advantage of these molecules are, they are biomolecules, but so they self-assemble and they have high mechanical properties. So their Young modulus and bending rigidity actually is comparable to steel, right? And they have thermal and pressure stability. So, and then they form material with these different sizes and morphology depending on their nature. So here is the deep ultraviolet resonance Raman data actually gathered by our collaborator. And as you can see that there is a characteristic amide bond that showed that they are beta sheet rich. That means that they stack together through hydrogen bonding. And also this is measured value of their Young's modulus, 1.8 gigapascal, measured using atomic force microscopy. And so using AFM, you can see the size. So you can get the width and length, but one doesn't know how many monomers are inside. And based on that, you can calculate their Young's modulus. So now this is a painstaking job. So what we wanted to do, can we create virtual libraries of these molecules and tell our experimental colleagues which of these would have interesting material properties, instead of they try one by one all of them, which is very expensive and time-consuming process. But in order to do that, first we need to check, can we reproduce what they see? So what we did, we had these structures of our different monomers, 60, 80, 120, we even have bigger model. We looked at the structural properties. I mentioned something called beta sheet. As you can see that all these structures, they are beta sheet rich, almost like 90%. And then, so we calculate their fibril twist, interstrand distance, interstrand angle, periodicity, and all that. And then we calculate their Young's modulus. So in this table, as you can see that how their Young's modulus vary as the function of their, like the size of the monomers. So as you can see that when you reach to 80 of these monomers, Young's modulus is 6 gigapascal. When you go to 120, it's 4.5 gigapascal. So that means starts converging. And then our collaborator, he used nano indentation AFM technique and measured their Young modulus at 1.8 gigapascal. Now you can say that there is a big difference between 4.5 and 1.8. But there is another group at ETH in Switzerland. They use a different AFM technique called peak force quantitative AFM, and they measured 3.2 gigapascal, which was much closer to our value. So you can see that even based on the which experiment AFM technique one uses, there is a big difference in the measured values of Young's modulus. So what we did now, we took another peptide and then which was A beta 42, as you can see that, and then it has totally different morphology. It looks like S. And then we also calculated just Young's modulus and then compared with the measured value. And so the long-term goal was here that, okay, can we come up with these, simple models and we can predict the structures of these molecules. So like that means we can we design rules based on the engagement. And so the goal was like we study their interactions, how the like non-covalent interactions like hydrophobic and hydrogen bonding, they contribute to the aggregation or the assembly of these molecules. And then we use those rules of engagement, and then we increase their density, like use these simple models, and we can probably predict their structure. So that we tried. That was a long-standing goal 13 years ago. And that's how it works in science sometime. And 13 years down the line, it's still a goal. We still don't have a model. We cannot predict their structure. So this is a big question. And we have reviewed and all these some of these results in these different papers. And so I will end up with this statement by the economists. It's in the real world, this could eventually mean that most chemical experiments are conducted inside the silicon of chips instead of glassware of labs. Turn off the Bunsen burner, it will not be wanted in 10 years. So that was in 1998, by the way, and then it's still needed, it's still used, all right? So I hope that I managed to convince you by just sitting in front of computer and using silicon chips, we can do some very useful chemistry. And these are all the collaborators and my group members who did all the work. And then it has been consistently funded by NSF. And I would like to thank you for your attention, and I would be we'll be happy to take your questions.
Thank you, Rajeev. Thank you. I'm going to stop the recording now to take the questions.
Okay.